Dr. Fraser Hof. I know if you've come to these meetings before, Fraser has spoken to us before, so it's nice to see you back again. Thank you for giving up your time to speak to us. Fraser is a chemistry professor at UVic since 2005. Fraser has been, was one of the first recipients of research funding from the Westcoast Motorcycle Ride to Live, and that was back in 2011?
Yes.
That was back in 2011. You were the first grant, actually, for the West Coast Motorcycle Ride to Live. So has been doing amazing research through that grant that leveraged a lot of other great things that he was doing. And then he also gave back to Island Prostate Centre by sitting on the advisory, the granting committee that hands out the funding for that event. So thank you for giving back your time to help with that. He runs a group of research students tackling prostate cancer treatment and diagnostics. And earlier this year, I know Red, you attended, he also gave us a great tour of his facility, which I know he is willing to do again if we wanted to organize it, because it's amazing what they're doing in the lab up there at UVic. So if you're interested, let me know, because I know there's others that would be interested in doing that again. And Fraser's just a really super nice guy. So Fraser, thank you for coming. Nice to see you again. And the floor is yours.
That's a lot to live up to. Thanks very much, Leanne. Just by way of introducing myself, I'm born and raised in Alberta, came out to the West Coast, as Leanne said, 11 years ago. I've been working in the area of chemistry, and one of the things I hope to be able to do today is kind of demystify a little bit what it is that we're doing, and just show you how researchers interact with each other, how we interact with students, how we interact with the community, and ultimately with patients. And so another reason I love coming back to these groups again and again is that it's also a chance for me to learn from you. There's a big gulf between research and patients at times, and I think the more that we get together like this, the more I get to learn back from you, the better the research becomes. And so I look forward to, I believe, lots of time for questions, put it that way, and I will not snow you under with chemistry, because I want you to think I'm a nice person. It's really not the point of me coming here. So this is really just to give you an overview and some context and show you how we think about the topic tonight that I've chosen is how we think about castrate-resistant prostate cancer as chemists who think about molecules and who wonder what we can do about castrate-resistant prostate cancer. So in a general sense, I'll just start with a couple of quick topics, talking about exactly how research ideas arise, how they can turn into progress for patients, and an overview of castrate-resistant prostate cancer, again, from the point of view of the researcher, and with a few kind of general definitions and resources that I think you might find useful if you're interested in reading more into this. So the first example that I use here is nothing to do with research in my own lab, but it's just a great example from other cancer research. It involves a form of leukemia, so bear with me for a second. It's really a general lesson in how there are control switches that control whether your cells are growing or not growing. Okay, so we've all heard of hormones, and we understand that these are molecules that circulate naturally in your body, and one of the things that they do is to trigger growth. Okay, so in order for them to do that, they have to bind to some receptor that's another molecule in the cells. So we can picture this little thing here with a pocket. We can call that a receptor. We can call that a hormone, and you can see when the hormone binds, this thing has turned green, therefore this is off, and with the hormone binding, we call this an on state. And with normal, in this case, growth of blood cells, because we're talking about leukemia, there's some amount of growth, and then there's some amount of regulation backwards, and so you have kind of a regular steady state. You need cells to grow, but not too many. What's pictured down here, though, is actually what happens when a mistake happens, when damage happens to the receptor. And so I've just kind of cartooned on here a glommed-on extra fragment of molecule. this is a broken switch. So this receptor now is stuck in an on position always. And the off and the balancing act doesn't work. The hormones are no longer required for the cell growth, and so we have this runaway growth and replication. Now this particular example is from a form of leukemia called acute myeloid leukemia. The reason I choose it as an example is that it's a poster child for what we hope to achieve with castrate-resistant prostate cancer. It's a fantastic success story. Before this pathway was understood to be causing the runaway growth, so before that basic knowledge was in hand, the only treatment for this form of leukemia was surgery that was very invasive in public bone marrow transplant, and the success rates were very low. Okay, so in the prostate cancer community, I think it's a regular piece of public information, and we discuss how good your outcomes are. In this particular form of leukemia, even after bone marrow transplant, they had something like a 30% survival rate after 5 years, and in the long term, worse than that. So with the understanding of exactly which switches are going wrong, and the idea then that you could block those switches with a drug, we went from 1993, the first molecules that could block that busted switch. 1996, the first creation of this drug, which is called imatinib, you might have heard of it, also called Gleevec, in the news. So the first ever creation of it was in 1996, and it showed very promising results in testing in cancer cells. And 5 years later, it was approved, and it's still used today, and relatives of it are used today. And for this pill, which is not invasive at all and is very easy to take and has very few side effects, now the numbers are 85, 90, 95% cure rate for this particular form of leukemia. So this offers us hope that specific knowledge of busted switches allows the chemists to come in, make drugs that block those switches, and have a really profound impact on the care of aggressive disease. A caution here is that at the time of this 1996 to 2001 advance, that was the all-time world record for the fastest approval of a drug. And it has since been broken by one relative, but this is not typical. Okay, so our normal timelines we think about here are more like 10 to 12 years, and I'll talk a little bit about exactly what's involved in that. But nevertheless, to give you a little bit of a context for these things, I just show here something that I actually show in a class that I teach at the university that's all about drug chemistry. And I want to point out that a lot of new ideas are floated, constantly generated by different researchers using different approaches. And I'll give you some examples of those later. And then right here in the middle, for me, this is where the rubber hits the road. So lots of good chemists, biologists, biochemists, cancer researchers of all stripes are involved in generating new ideas here. In order to arrive at a drug, a chemist is the person who joins the team, normally after the original ideas are floated. And they make molecules, they test those molecules, they optimize those molecules, and they eventually get them through this stage, which is from blocking molecules in a test tube to blocking molecules in an animal to hopefully having those blocking molecules actually working properly in human beings. And then these phases here are where we go through progressively more stringent testing in humans. And then finally, government approval and follow-up testing. So there's a large gap between universities and all of our academic collaborators, which is the world that I live in, and the pharma companies that actually have the resources to bring these things to market. I think discussing the specific numbers or debating the specific numbers isn't very valuable, but the cost of bringing a drug to market is definitely something north of $1 billion. Okay, and some people say more, and some people say a little less, but boy, it ain't in the budgets of these institutions. That's for sure. Okay, so just to kind of, it's important for me that you understand our role, but also our limitations. We have ideas and we do a lot of interesting work in this space, and the idea is that we're going to hand it off at some point to private enterprise. or to other consortia. Now there's something that has come about in BC, a really interesting made in BC solution just in the last less than 10 years, which fills this gap. And so I'll just highlight very briefly, there's an institution called the Centre for Drug Research and Development. It's in Vancouver, and it's located on campus at UBC, but it's not a part of UBC. It's really an independent institution, and their entire mission is to fill the gap between universities and collaborators from all across Canada and all across the world, that gap between new ideas and promising data and actual commercialization. So they have a full facility staffed with experts who come from pharma in every different division of science you can imagine. They run these kinds of Dragon's Den kinds of not competitions, but if you want to work with them, you have to go through a Dragon's Den gauntlet. Except, on Dragon's Den on TV, they have 4 people scolding you and telling you your idea is terrible. And when I've done this at the CDRD, there was a conference room with 18 chairs, and there were 24 people in it. And every one of them is scolding you and every one of them is an expert in their own different area. And at the end, they say, "You did great." And so it's humbling, but also really stimulating and really interesting. And so through them, we've been able to partner with multiple private sector drug companies, because it's all about them building partnerships for different bits and pieces of our research. Is there a question?
How do you deal with the intellectual property ownership?
So the CDRD and the university and pharma, they all have agreements. The intellectual property is sorted out ahead of time. And in most of their agreements, the intellectual property resides completely with the universities and the inventors. If the CDRD staff themselves make an important invention as an addition to our invention, then fair is fair, and they're going to get a piece of the intellectual property. Most of their agreements involve pharma not taking any intellectual property. But they do have access to data that they find valuable for their own programs. That's at least one form of agreement, and we have had other forms of agreement with them too. There's one in particular we're working on right now where the pharma partner will maintain intellectual property that they have arrived at the partnership with. So it's important and it's done up front, is the short answer. Okay, that's a little bit of context. Let's talk about castrate-resistant prostate cancer from the point of view of the researcher and especially the molecules. So there is actually no castration in castrate-resistant prostate cancer.
Change the name, okay.
Yeah, I know they should change the name. But it used to be called more commonly hormone-refractory prostate cancer. But that name was dropped because as newer hormonal treatments have arrived on the scene, they've actually shown some usefulness in castrate-resistant prostate cancer. Some, but not others, but you can't call it hormone-resistant prostate cancer anymore. And so yeah, that's the name, okay? It is cancer that's progressing normally through rising PSA as a main marker, despite the treatment with anti-androgen, as anti-hormone therapies. And most often CRPC is metastatic disease. So as researchers, we focus on CRPC because this is the most difficult challenge facing prostate cancer patients right now. So this is a form of the disease that's more aggressive and for which there are fewer treatments available. Now, I think the first time I spoke to this group was in 2011, and at the time I had this 2010 guidelines, which I set up and talked about a little bit. So these guidelines come from the Canadian Urologic Association Journal, and they're published periodically, not every year, and they're specific for Canada, and they talk about what the state of the art is for treatment of castrate-resistant prostate cancer. And so I was looking over this today, and I found in the 2010 guidelines these statements, which were really reflecting the state of the art then, and I think there's actually been really good news, really exciting news in the last 5 years. So just in the time that I've been coming to these meetings, we can go from basically supportive care, palliation and maintenance of quality of life as the main objectives for CRPC treatment 6 years ago, to now multiple agents that have shown real improvements in the duration of survival of the patients. And they include agents of multiple different classes, including these two, which I'll talk about a little bit more. These are modern hormonal therapies of different kinds, or hormone-related therapies. Docetaxel and cabazitaxel, these are chemotherapies that have shown efficacy. And radium-223 is, of course, a chemical version of radiotherapy, radiation therapy, which is also showing efficacy. And you can read these guidelines. I think I can get these links back to the group easily enough, right? These are freely available online, and you can read the guidelines and see in technical jargon, what is recommended. As a researcher who speaks both languages a little bit, I'm always happy to help patients interpret things that they find in technical literature. I should have said this right up front, though. I'm the wrong kind of doctor, right? So I'm a PhD in chemistry. I will never give you medical advice, right? Because I shouldn't. But if I can help you understand something that you read somewhere else, I would be happy to do so. Okay. My wife calls me the wrong kind of doctor also. For other reasons. So what have we been doing lately? And in particular, how do we find out about molecules that turn easily treatable prostate cancer into castrate-resistant prostate cancer, and what are we doing about it? Okay, so let's get anatomical just for a second. So we know that cancer is the growth of cells that is uncontrolled in a certain tissue. Within those cells, so this thing that's stained blue in this particular nice fluorescent image, this is the nucleus of the cell, and within the nucleus is DNA. So we recognize this picture from high school textbooks. Those are nice double helix. DNA is compacted into chromosomes. You might even recognize that picture too. This is the area of the cell where my lab does most of our chemical research. All of this genetic information represents data, a program for how the cell is going to work. It's not always turned on. Okay, so we're back now to the idea of switches. And in particular, we're wondering about which switches turn the genetic program on. Just simply, which switches say to the cell, yes, you should read this program, produce the products of that program, and therefore fuel the growth of the cell. So there are many such switches in different kinds of cells. In castrate-resistant prostate cancer, the switch that we care most about is the androgen receptor. So we're back to the word androgen. This is just a word that means male sex hormone in general. The androgen receptor here I've cartooned as a simple chunk. This is a protein with amazing power, diverse functions, but fundamentally what it does is that it binds down on the DNA, and when testosterone or another androgen binds to it, so there's T for testosterone binding down here at the binding site, that turns it on, and then away it goes. And the program is read out, so it's actually physically on the program of DNA and chugging out information, and in this case, driving growth aggressively. Okay? Normally, only when testosterone is binding, so we're back to the idea here of a hormone binding to a receptor in order to fuel growth. Now, this is the androgen receptor, is the switch that matters in CRPC. So let's look at it a little more closely and see in general how this behaves in prostate cancer. So we're back to the same cartoon. We have regions down here that bind to DNA, and the gray box here is the region that actually is binding to the hormone. So that binding domain, I have the T pictured there. And there are a couple of drugs I think will be familiar to any of you who've been reading. We can talk about a little bit more. There's abiraterone. This is a drug used for the treatment of prostate cancer at multiple stages. This is a drug that actually stops the creation of testosterone in your body in the first place. So it blocks something upstream so that testosterone is not made in the first place. Okay. There's another drug, another next generation hormonal therapy called enzalutamide. One of the mechanisms of that is that it actually blocks the testosterone binding to its binding pocket. So basically it binds in the binding pocket and it keeps the testosterone from binding there. And in that way it shuts down hormone signaling. This plus is written here because enzalutamide is actually shown to be a pretty clever drug and that it also works by multiple other pathways. But this is definitely one of them by which it is functioning. Okay, so these are ways of treating prostate cancer. You basically are shutting down testosterone signaling one way or another. Now, here's a real problem that in the same way that I referred to a molecular switch being broken and stuck on in the earlier leukemia example, the same sorts of breaks can occur in the androgen receptor. So the cartoons here now are useful because what they show is that the four most common broken forms of receptor are actually missing that binding pocket altogether. So the binding pocket where the hormone binds is no longer present, and these things are just kind of active all the time. So it's another version of a switch that's stuck in an on position. What's really a problem from the point of view of the chemist, what does a chemist do? We can make molecules that bind in binding pockets. So we have a physical problem here, and that's that the main well-known binding pockets are just absent from these things. And so it's quite hard to think about how to make drugs that can block CRPC, that can help treat CRPC when the most well-known binding region is simply deleted from that receptor. So that leads us to think differently, and other cancer researchers think differently. So if we just take one example of them and think about how it actually acts in a cell, that I have to make a confession here now, which is that nothing in a cell is as simple as one receptor fuel, with one broken switch fuels all of the bad growth, and then you can cure it that easily. The real truth of it is that this is actually a real network map of all of the different switches interacting in one particular pathway in the cell. Now, the androgen receptor is one key switch. What we hope to do is identify some other switch by which we can block indirectly the same signaling cascade, the same pathway of growth. And this is legit. This is a real pathway by which lots of cancer drugs have been developed. But you have to figure out what that candidate, what that drug target would be in the first place, or that would allow you to just wipe everything out without directly targeting the androgen receptor. So I'm going to kind of jump ahead here, years worth of work and pondering and thinking to just some things that we've been doing lately together with collaborators in this area. So our key collaborator these days on these projects is Dr. Cheryl Helgeson. She's a prostate cancer expert at the BC Cancer Agency labs in Vancouver. We also work with BC Cancer Agency folks here in Victoria on other projects. But Dr. Helgeson's lab actually found some interesting data that I'll run through here quickly and that we've been working together on in the last year. So in this instance, she was able to take cells from one prostate cancer patient from two different biopsies, one of which was metastatic and one was non-metastatic disease, and then really just look for differences. So the question is, what's the switch? What's making the disease metastatic instead of non-metastatic? And she had a list of candidates. So this is a rational guided approach here. And the list of candidates included many proteins that my lab was already studying. And her lab came up with this very striking result. So the little black bars here are non-metastatic disease, and the gray bars simply show the levels that these proteins jump up to in metastatic disease. And it basically is flat except for this one. Okay? So this is a protein called CBX2. Forgive the jargon, I could give you the whole name of it, but it wouldn't make you love me. Okay? So CBX2 is a protein, it's another molecular switch. And it came out of her study on this one prostate cancer patient sample as something that was really noticeably higher in the metastatic patient, in the metastatic case, yes.
So were these two biopsies on different parts of the body?
Yes. I think they might have actually been from different time points in the same patient, more so than different parts of the body.
Because I always assumed metastatic disease was just the same thing, moved elsewhere, no?
No, but normally, metastatic disease is started from the same point, but in order for it to become aggressive, its molecular signatures have changed in order to drive it to go somewhere else in the body.
Okay.
So yes and no, if you follow. Okay, so CBX2 is a candidate, but you never know what you're going to get from a single patient sample. Now, here I want to highlight something that might have touched on you in the past, but it's definitely touched on our research, and that's tumor tissue repositories. Okay, so patients can, on occasion, they can be offered the option of donating tissue to tumor tissue repositories. And so in this instance, biopsy samples are collected and cataloged, and they're tied to you, the patient, and your characteristics, but not your name, of course. Nothing that identifies you. Those samples are studied in every which way you can imagine a modern biochemical analysis being done. And all of that data is cataloged. So we can look at genomics and transcriptomics and all of these omics words basically just mean like shotgun measurements of everything you can imagine. So every gene sequence, every protein level, everything. That's omics. At the same time, the doctors are feeding the patient outcomes and progression back into the database so that what you end up with is a vast database, many thousands of samples normally, where a patient outcome and characteristics are tied to real biochemical information. Now, this is a powerful tool for prospecting. You know, as researchers were looking for a needle in a haystack, remember the ridiculous diagram I showed before with all of those little boxes? We don't know from scratch what we should be targeting, but data from these databases can help us. We can go back in after the fact and say, hey, what about CBX2? Now that we have this lead, we have this idea. And so Cheryl did that. And she actually did it in a variety of different cohorts. And I'll just highlight here, you know, so her idea was that CBX2 is a marker of metastatic cancer. And what she found in 3 different prostate cancer data sets was that same high amplification of CBX2. So this is a little bit, but 7.8 and 9.2 times higher. This is really full-blown high levels of CBX2. And now we're looking at dozens of patients. And in other diseases, you know, she's getting up to hundreds of patients, but she's really a prostate cancer expert, and she's focused on the prostate when she does her work. And further, she can go and look at specific data sets where you can follow, in this case, 140 patients at Memorial Sloan Kettering Hospital. So that's in New York City. We're also participating in a study of this sort. Good news, so the bad news is that if you have high levels of CBX2, you have a very bad prognosis from among these advanced prostate cancer patients. The good news is that it's relatively rare. So only 8 patients represent this trend line of patients failing treatment and having disease recurrence. And 132 out of the 140 actually are this trend line with very good outcomes and good survival, disease-free overall. Dan?
These data sets, are they in the public domain?
So these data sets are in the public domain. They are difficult to use. But there's a lot of open science in this area. So there's one called OncoMine, which the idea is you're mining the database of oncology. There's one called Cosmic, which is an acronym COSMIC, Cosmic. I would happily kind of dig through things with you at some point to the extent that I can help. But yeah, they're public domain, and a lot of public science happens here. Okay, so this picture isn't coming out very well now, but basically at this point, so Cheryl has learned from an individual patient what might be important, and she's learned from various databases of really valuable donated patient samples that this is a real deal, that it's important for hundreds of patients. And she has one more trick up her sleeve, and it's these aren't cartoons, these are microscope slides. This is untreated castrate-resistant prostate cancer cell growth. These are petri dish kind of microscope size. This is all cells, okay? And here, when she can actually manipulate the cells to reduce the level of CBX2, she sees very few cells. Okay? And actually, the green line, if you're a stickler over here, this is the same data represented in a way you can see. So the green line is cells dying, and these other lines are the untreated cells growing happily. So that's like a biochemical trick that Cheryl can pull, but it still isn't a treatment. So at a certain point, a year and a half or so ago, Cheryl, she contacted me. My group had done some work on related targets. She found out about it. She contacted me. I contacted her. We very happily started thinking of different ideas because so I'm the chemist. So she's the ideas person, and the chemist tries to make the ideas actually work, actually real, actually testable. Okay, so the chemist has to make a drug. And here's where I'll kind of get out of the way and spare you all of that chemistry. Remember, I promised to spare you chemistry, but just a couple of examples of how these things are working. So here's a chemical structure. By all means, ask me to interpret it afterwards if you want. But that's a particular structure that my lab invented and made, and it had a first-of-a-kind, it was a first-of-a-kind inhibitor, but it had disappointing activity when we did the studies in cells. So it was very good in the test tube, less good in the prostate cancer cells. Now, actually, Dan, here's an example of scientific openness. So we published this work as it was. We didn't file any intellectual property on it because we thought, well, this is not going to be a good drug, but we want the world to know about it. And another lab, University of North Carolina, actually came along, and you can see here in red, this little group, this little group, that little group, that little group. Even if you don't understand chemical structures, what you can see here is that most of this is the same, but they made some clever substitutions. And they found something that was 30 times more potent in cells. And now you can see the microscope images. This is just the prostate cancer cells growing in their normal, uncontrolled way. And you can see here, cell, 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 cell. Most of the cells are killed off by this analog that the group has made. And on the one hand, one of my students kind of cursed and swore and said, they're competing with us. But on the other hand, actually, I just visited there less than 2 weeks ago, so they invited me down. They know about our work in the area. There's a lot of scientific openness that helps things move forward. And it's to the credit of this lab. We're very open about things, and we've swapped ideas and continue to work on these things together. So I much prefer the latter viewpoint of, let's say, friendly competition and sharing ideas rather than that one student's viewpoint, which was to shake his fist angrily and worry about his ownership of things. And here, this is the last slide I'm going to show you of any sort of scientific data, and it's simply that with Cheryl now and with the new student, Natasha Milosevic, no fist shaking from Natasha, that we have this really exciting result recently. So here is Cheryl's modern results on castrate-resistant prostate cancer. And here's a chemical that Natasha invented together with other students helping her out that showed this curve. Now this curve really, if you look here, this is the important thing, viable cell count. So this is tens of thousands of cells in the petri dish. This is dead, like just deader than dead. So these castrate-resistant prostate cancer cells are very aggressively being killed off. It's not a subtle effect. And so this is something now that we're pursuing aggressively with some pretty major funding together with Cheryl at the BC Cancer Agency. So okay, let me back up a little bit from the graphs and everything and just talk a little bit more about context and the value that we find in this community as researchers. So a normal acknowledgement slide, if I were at a scientific conference, would just have a list of funders, people who gave me research grants along the way. Now, the Ride to Live would be prominent among them. It always is. So this is the Westcoast Ride to Live that Leanne mentioned. It's a local charity. Many of you know everything about it. And if you don't, you can learn about it in this room for sure. Okay. So this is a little different than that slide, because what I wanted to show was that an early PhD student of mine had funding, an award from Ride to Live, in the subsequent year, he got an added bit of funding from Prostate Cancer Foundation of British Columbia. So that's a Vancouver-based charity that has a totally independent existence. And Kevin, he actually won the Pioneer Award in that competition, which is the award for the top student or postdoctoral researcher in the province of BC, which is pretty cool, and there's a crystal trophy that goes with it. And Kevin has gone on, he graduated with his PhD, and he's gone on to this very prestigious postdoctoral fellowship funded by EMBO, which is the European Molecular Biology Organization, at an institute in Germany. So I hope you see the role that Right to Live played as the seed for Kevin's personal progression as a cancer researcher. Natasha, she won funding from the Right to Live in 2014. This is actually, I don't think Leanne told you about this, but she just 2-3 weeks ago got word from the Prostate Cancer Foundation of BC that she got a fellowship, and they give her the Pioneer Award as the top student in British Columbia.
We were there when she got the award.
Oh, you were there?
That was in Vancouver, yeah.
I was in North Carolina.
Okay.
Yeah, that's why but yeah, so that's fantastic for her. Also, now this is the project that started us on the path of chasing down this target, this whole branch of work, let's say. And with Cheryl Helgeson, we have this major funding from the Canadian Cancer Society to pursue this, and that's what we've been doing over the last little while. This is just one little snippet of the sorts of things that we do, and I thought it might be nicer than just kind of showing, oh yeah, and people pay for this stuff. One thing I want to point out is that whenever any of this money is awarded, it only goes to 2 things ever. Okay? And one of them is paying the direct costs of what we buy to do the experiments within the lab, because we have to buy that stuff. So the chemicals and the reagents and the cells and so on. And the other thing is that we have to pay our researchers a salary, a stipend, in order to be able to live in Victoria. And so the one thing that never happens is that I get any money out of it at all. Right? And that's common across all of research. Researchers don't get paid by the research grants that they get. And I should say that's universally true in Canada. In other countries, that's really not true. And in America, there's a lot of salaried researchers who draw their salaries from research grants, and that's cool, that's how they live. But for us, this is really about like students and training and just paying the bare costs of what we're doing in the lab. So all of this happens in a network of collaborations, and we have here a list of different collaborators we've worked with on different projects. So I highlight the Centre for Drug Research and Development, the BC Cancer Agency, as 2 made in BC partners that we work with a lot on a lot of different projects. I like this slide because this is actually every student who worked for me between 2005 and 2015. I just haven't updated it this year. There's another 15 students I need to add to that list. I need to make the font smaller. But I just want to remind everybody that this, we're a university, we train people, and our main mission is creating new knowledge and putting that knowledge into the brains of bright young students who go off and do great things. And finally, I'll just point out that there's Kevin. He's a former Right to Live award winner, who we guilted into riding in the back of a motorcycle once. There's a picture of me in the back of a motorcycle that I'm not going to share. And actually, here's the lab tour. So actually, already it was mentioned that I like to give lab tours, and I was going to finish with that. Here's the first lab tour we ever did. These are 3 Right to Live guys who are Blue Angels. They're retired police officers. They made me pose in the foreground holding a chemical up to look pretty. But really, this is just up here to say that I'm happy at any time to arrange lab tours. You can see what we're doing. You can meet the students. We can show you around what we're doing at the university, and that's easy to arrange through Leanne or Dan or anybody in small groups or big groups. I just think it's a really important thing that you can do for us. Whenever you come to visit the lab, the students get really excited and motivated afterwards. So that would be a great thing if we could keep doing that. And that's all. That's all I have to say. Thank you so much for having me.
I wonder if you just clarify one thing for me. see if I heard this right. You were quite excited about what this woman discovered with CBX2. You said she had 140 patients, of which 8 of them had high CBX2. I didn't get the disconnect there. I didn't get the connect. The 140 patients were cancer patients who she tested, and only 8 of those. That's where you said it was relatively small, the number that have CBX2 on it.
Yeah, so first of all, she didn't test them. That was another example of database data from patient-donated samples.
But they were prostate cancer patients.
So those were prostate cancer patients who had been diagnosed with prostate adenocarcinoma, which is not CRPC. So that's something lower grade, earlier stage, and not specifically defined as CRPC. And so they were just looking at that 140 patient set of, let's say, more general, probably say that's very more general prostate cancer patients. They were looking for molecular signatures. And they weren't looking specifically for CBX2. They collected thousands of bits of data and plugged them into a database. So Cheryl's contribution is to have the inventive idea that maybe CBX2 is a key marker, and she saw a few clues of that, and then she can go back to the database, to that open science data set for those 140 patients of general prostate cancer, and then find 8 of them have the high levels, and those 8 are the ones who died rapidly and resisted all treatments. So it's an after-the-fact investigation, and that's how most of this goes.
Maybe flip back to that slide and explain that graph, how to read it.
Yeah. So, so this is the fraction of people in the population between 0 and 1. So 1 is all of them and 0 is none of them. Okay, and this is all retrospective after they've gotten the best standard of care. There's nothing experimental going on with them. Best standard of care for their conditions with their physicians. Okay, they just divide them into those that had this high marker, which we'll call the broken switch, and there were only 8 of them, and you can see here, you can see them actually succumbing, disease-free survival. This means, so 1 died, another died, another died, another died, and then the rest of them died, and this endpoint here is within 5 years. So these 8 patients have died rapidly, and all of them died within 5 years. The 132 patients are represented here, and the fraction of them that are surviving is 65% by the end of by the very long term end of the time frames. This is well over 10 years. Okay, and so most of them who had normal levels of CBX2 are surviving disease-free, completely disease-free.
If that's disease-free survival, then someone could fall off the graph if they kept the disease as well, right? They didn't necessarily die.
Yeah, if they have a recurrence, you're right. And so these graphs are done in different ways. Sometimes this is just disease-free, and sometimes it's progression to death. Oh, sorry, when I say succumbing, I mean, and the disease is recurring.
Thanks. That's a big difference for us.
Of course. But of course, none of you have this particular marker. This isn't used in a diagnostic way.
But if we're going to read that kind of diagram for our own understanding, the graph is useful.
Of course.
Yeah.
On the tour that I took, I guess it was about a year ago, it was a very good tour. One of the things you mentioned is that you were getting a medical doctor to be associated with the project work. How's that working out?
So we have, we have medical doctors associated with lots of our projects. And I'm trying to remember exactly which one you're asking about.
Yeah, it was one that was right on site. Was you pointed out an office that he or she would be occupying and closely associated with the work the students were doing?
I don't actually know what you're referring to. One of our key collaborators is a medical doctor who's in the Netherlands, where I just spent a lot of time. But I don't know exactly what you're talking
Yeah, my recollection is that the medical doctor was going to be on site part-time.
No,
No, okay.
no. But we have postdoctoral fellows who have PhDs who work in the lab. And I wonder if that was the source of some confusion.
Yeah, could be.
So these are full-time researchers who already have PhDs and they come and join the research team. Maybe we can plumb the depths of our memories together later and try to figure that out.
Or see if I can find the video that I took that night.
Evidence.
Evidence.
I just want to make sure I understand the CRPC correctly, and that is, if you're on permanent androgen therapy and yet your PSA count is still going up, that's caused by CRPC, am I right?
That's one of the definitions of CRPC, but that doesn't define whether it's metastatic or not. Right?
Yes,
That's the,
of course. Yeah.
Yes, in the back.
How fast does the cells die off once the testosterone's been removed?
Do you mean in your body?
Yes.
Yeah, that's, you know what, I don't know. Ask me that question about Petri dishes and I can show you a graph with days and I can tell you that we see big effects after 6 days, really profound effects after 6 days, but if you look after 2 days, it kind of looks like not much is going on. Like there's a lag in this particular set of experiments that we do. And that's all I should say about it, because I just shouldn't speculate about what's going on in your body during your treatment. Yeah, well, for sure, yeah.
Because I've been on the for 20 months, and for the last 6, my PSA is down at undetectable amounts.
That's good. I'm not a medical doctor, but I know that's good. Yeah, it's great.
So something like this began to look quite promising. What would be the stages? Right now you're doing it in a test tube or a dish, Petri dish. What would be the stages for something like this to actually get out to real guys? And how long might that take?
Yeah, so Petri dish, mice, rats, and then normally some experiments with larger animals, including dogs and monkeys, but those are just to make sure that the drug is safe. You're rarely doing a prostate cancer model in a larger animal. Okay? So there you do safety trials, and you have to do a lot of safety studies before you ever give a drug to a human being. And then the first phase of testing of drugs in human beings is again mainly focused on safety. So they're watching you like a hawk for adverse effects, fevers, chills, nausea, heart irregularities. Safety, 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 safety. It's a necessary evil. You have to test a drug in a human. Some human being has to be the first person to take a drug. Okay? And those people are watched in small numbers very carefully to make sure that the drug is as safe as possible. And depending on the disease, it might even just be a healthy person that they're doing that safety study. Normally in cancer, they do use sick patients. You can just watch them for safety profiles carefully. If that turns out all right, then you can start doing bigger studies with more patients in multiple different hospitals around the world and start to really study the disease. Okay? So the how long does it take, is really 10 to 12 years time frame, roughly, from molecule to fully approved for use in humans. And there's a huge rate of attrition. There are lots of good ideas that end up not working in human beings, not working in mice. And that's why it's so expensive also, is the attrition rate.
I've got a very basic question about cancer research. It seems as though each cancer researcher is looking at a particular type of cancer. Is there anyone looking at the whole thing and seeing if there's any way that there's a mechanism that solves all cancers?
So, cancer is not one disease. Cancer is hundreds of diseases, and each one of them has a different molecular mechanism that causes the uncontrolled growth. So I don't just mean that breast cancer is different than prostate cancer is different than liver cancer. there are multiple kinds of breast cancer. Each one of them is a totally unique disease that arises for a totally different reason. So the way that I think about this is that each of those cancers needs to have a cure identified. This is a long slog. There are no magic bullets. There can't be magic bullets because it's like saying, well, what if there was one drug that would cure influenza and salmonella? You know, and one's a virus and one's a bacterium, and they have really nothing to do with each other, and it would be nice, but unlikely. Okay? So people look at overall themes in cancer and find that one kind of drug or one kind of pathway is important here, there, and there, and there, and there. So it's not all the most pessimistic thing in the world. And I have other slides I've shown in other presentations that show, you know, really dramatic, significant improvements over time in the survival of patients for lots of different kinds of cancer. But that sort of leap that I showed for imatinib for acute myeloid leukemia, that's what we're chasing. You know? It's nice to live an incremental amount of time longer, but what we really want is to go from good, effective treatments to just cure that disease, you know, for 85 or 90% of the patients. And that's what a lot of researchers have focused on these days.
You mentioned earlier about the turning switches on and off. Does nanorobots come into any play anything in that?
So there's no such thing really as a nanorobot. There are molecules that do certain functions at the molecular scale. And it sounds really cool if you call them nanorobots. But they're not made of metal and they don't have electronics in them. But just to give a cool example, there are molecules used for the treatment of eye conditions that only turn on and do their thing when a light is shone on them. So you can give the person an intravenous injection of this drug. It goes everywhere in their body, all their blood vessels. You don't want it to be active everywhere, and so just where you shine light, you can shine light onto the retina through the eyeball. Turns on the action of that thing there. Okay, so in medicinal chemistry, we call that a light-activated drug, or a phototherapy. In Scientific American, they call it a nanorobot, you know, zapping your eye with a laser. And so there's not a lot of truth in the nanorobots world, although it's useful to think about molecules doing work. You can program molecules to do cool things. They're just not robots. So with that said, those sorts of programmed molecules, they are particularly interesting in areas like skin cancer, for the same reason you can turn it on with light. Well, if your skin cancer is shallow and it's right there, and you can put the drug everywhere, but it only kills the cancer where you're shining the light on the cancer, that's a nice idea. Right? You're making the molecule do work where you turn it on. Those sorts of things in prostate cancer, I have not seen much in the way of progress there. The prostate is deep, it's relatively hard to get at. Light is not going to do it. And so you have to think of other tricks in order to make progress there.
So if the different cancers will obviously have different triggers for the cells to start to go crazy and divide and such. And so that must be a horrendous challenge to try and find what all those different triggers are.
Yes. So prostate cancer is actually relatively simple. There are relatively few of those triggers in prostate cancer compared to, for example, leukemia. There are lots of varieties of leukemia and lymphoma, and each one of them makes up a small percentage of the overall number of cases. Prostate cancer is relatively well behaved in its progression, and that's why prostate cancer has such long survivorship compared to other cancers. And I'm not here to tell you that current therapies aren't good, because they're really, really good. And the progression of treatments that you'll get, whether it's surgery, radiation, chemotherapy, or any sort of the hormonal variety of therapies, they know when to use them, and they know how to use them, and they're very effective. And that it's a good thing relative to other cancers that you're in the prostate cancer community because of that relative simplicity. are relatively few things that go wrong in prostate cells.
Just a quick question. You kind of had mentioned in CBX2 how rare it was and how individual patients are. Do you find that hard as a researcher? Because I'm sure as a drug company, maybe correct me if I'm wrong, they're looking for more widespread use. Is that tough as a researcher, find the needle in the haystack and then somehow market it so that it's widespread use? Or?
Yeah, so the fact that it's tied so closely to metastatic castrate-resistant prostate cancer, so that's a rare disease, right? I mean, among prostate cancer patients, that's a relatively small proportion of them. So that's a strength. What the drug companies are very intensely interested in right now are treatments for metastatic castrate-resistant prostate cancer, because that's where there's a huge need. There are drugs that are showing some efficacy, and that's good news, but there's a lot of room for improvement, for like revolutionary advances in the treatment of that particular form of prostate cancer. So CBX2 is rare, but it's tied to metastatic castrate-resistant prostate cancer. Therefore, it's not a hard sell at all. And the drug companies are very interested in anything that looks like it could be a radical different approach to that disease.
Is there a biochemical reason why cancers manifest themselves later in life?
Yeah. So most, one of the number one risk factors for getting cancer is smoking. One of the other top risk factors is living a long time. Okay, and both of those things involve DNA damage accumulating. Okay, so smoking is a much more aggressive way to damage your DNA. Living a long time, DNA damage occurs naturally all the time in your body, and you have repair mechanisms in your body. It's a whole family. It's an even more complicated cartoon than the one that I showed. That's all about DNA repair. So your body's fighting a constant battle to repair DNA that's just degrading naturally on its own. And so over time, more and more DNA damage builds up. And it's not specific, right? But there are certain predictable accidental breaks that will happen that then lead to specific diseases. There are other risk factors we can talk about, but that's a big one.
Great. Thanks, Fraser. That was great.
So much.
I think